So I need you to know something about the cell cycle. And you may be aware, or did you talk a lot about the cell cycle in biology?
Yeah.
Okay, so this is review for most of you. Okay, so maybe we'll add a few things that aren't review. Many cells spend their life cycling between two different phases, mitosis and the non-dividing phase, which is interphase. And these are generally cells that need to be replenished and frequently include most cells that line body surfaces, like the skin, the lining of the digestive tract, the cells of the bone marrow. These are cells that are constantly dividing, okay? Especially red and white blood cells as they wear out. They're constantly dividing. Other cells never divide except under extraordinary circumstances. These include cells of the nervous system, skeletal muscle, heart muscle, and muscle that lines the digestive tract. These cells almost never divide. Now, there's an observation that we can make that's a very, very interesting observation. The types of cells that we get cancer in are the types of cells that are constantly dividing. We almost never get cancers of the brain, although you hear about them occasionally. They're very, very rare. We almost never get cancers of the muscle. We almost never get cancers of the bone. All of those types of cancer are very, very uncommon, but the types of cancer that we do see most commonly are cancers that occur in cells that are constantly dividing. What kind of a hypothesis might you come up with then to make that link there between cells that are constantly dividing and cancer? What does the process of constantly dividing make it possible to happen in those cells that would lead to cancer? If my cells are constantly dividing, I'm constantly duplicating my DNA, what kind of thing might happen in cells that I'm constantly duplicating my DNA? Yeah?
If a cell becomes cancerous, then it just duplicates the cancer cell.
Okay, that's one part of it. But if my DNA is constantly duplicating, what might happen to it when it's duplicating?
There's much more chance that a mutation might occur.
Yeah, I'm going to make a mistake, right? If I make a mistake in that duplication of my DNA, then I'm going to, and most mistakes don't cause bad problems to happen. It's only after I accumulate several mistakes that I start to get changes that lead to cancer. But the thing about this is that if I'm constantly dividing, I have a chance to make a mistake. My brain cells never divide. I don't get brain cancer. My muscle cells never divide. I don't get muscle cancer. My bone cells don't divide that much. I do get a little bit more bone cancer. But the skin cells are constantly dividing. I have all kinds of skin cancer. The cells inside my lungs, if I cause them to constantly divide because I'm smoking all the time, then I'm going to cause lung cancer. So, and one of the things that happens is that these cells don't normally make mistakes. These things don't normally make mistakes. But when we do things to our cells that wreck the repair mechanisms, then we allow this genetic damage to accumulate. So, for example, with lung cancer, the cells that line my lungs are constantly dividing, but they normally wouldn't cause mistakes in that cell division. But if I smoke, then I create a situation where those cells can't repair any mistakes that I make. And so any mistakes that I make are allowed to go on, and they get passed down to their daughter cells. Okay, so cigarette smoke is a good example. Certain types of viruses, the human papilloma virus causes cervical cancer. You've probably seen advertisements for the cervical cancer vaccine for human papillomavirus. But it causes a situation where the cervical cells can't repair the genetic mistakes that might happen during cell division. The sun damaging my skin. Those cells, the sun causes the cells to not have the ability to repair those mistakes. So even with all of these cells rapidly dividing, most of the time we wouldn't get cancer unless there was some environmental factor that went along with it. Now, there are certain types of cancer that are genetic. You inherit a tendency for these cells to get cancer. Breast cancer is a good example of that. Okay? So be aware that this is hugely, hugely, hugely important related to the development of cancer. Okay? If I screw this up, I get cancer. So it's important to understand which cells are constantly dividing and which cells aren't. It explains a lot about why we get certain types of cancer more than other types of cancer. So major phases of mitosis and interphase can be further broken down into a series of events called the cell cycle. Okay, no mystery there. And there are 4 major phases. Okay, so we have the M phase, which stands for mitosis, and this is when cell division takes place. We have the G1 phase. The G stands for gap, meaning the gap between when mitosis has occurred and when DNA synthesis will occur again. We have the S phase, and the S stands for synthesis. This is when the DNA replication will occur. And then we have the gap 2 phase, and this is when the machinery that are going to, the machines that are going to cause the cell division to occur will be created. Hopefully it makes sense that if I'm not going to divide, there's no reason for me to make the proteins that cause cell division to occur, right? If I am not going to divide, there's no reason for me to have those proteins in my cell that will create cell division. So I'm only going to create those cells or those proteins when I know that I'm actually going to undergo cell division. So once I've synthesized my DNA, once I've replicated that DNA, then I can make the proteins that will allow the cell division to occur. Okay, so this gap 2 phase is primarily for creating those proteins that are going to accomplish the cell division, and I don't need those if I'm not going to divide. In the G1 phase, I'm recovering from cell division. What do I have to recover from? What's happened in cell division that I need to recover from? If I go from this cell to these 2 other cells, what do I have to recover from?
Being ripped apart.
Yeah, I've lost half my stuff, right? I've got a bunch of stuff in here, and when I divide, I've lost half my stuff. Okay, now keep in mind the reason that we divide in the first place is because I want to avoid DNA overload, and I want to avoid problems with cell surface to volume ratio problems, right? So it's a good thing that I undergo cell division because now I've gotten to a smaller cell where I can more easily manage, but I'm going to grow and accomplish the functions that I need to do. So eventually I may need to divide again in order to maintain my size ratio. Okay? So we're going to create a diagram, and you can probably do this regardless of whether we're talking about it or not. You probably remember some of it. We're going to create a diagram, a cell cycle diagram. And it's going to be in a circle like that. And I'm going to have another circle in the middle here. You'll see why in a second. What's that?
Nothing.
Okay. Now, when we talk about interphase, you should recognize by now the prefix inter means between. Literally, this is the between phases. Okay? Interphase is the phase of a cell's life that's between cell division. And it used to be that when we first started looking at cells, we saw a few cells that had these things in them, right? These are chromosomes in cell division, right? This is what the chromosomes look like when we're in cell division. And then we saw a lot more cells that looked like this. Where all of these things were dispersed. And that's what the term for this is. Here I have condensed chromosomes. Here my chromosomes are dispersed. Okay, they're spread apart. Okay, and there were a lot more cells that looked like this than there were that looked like this. But these cells seemed to be doing something. Okay, it looked like they were active. It looked like they were doing something. Okay, they were dividing, but the anatomists assumed that when the cells were not in this phase, when they were in this phase, they weren't doing anything. Okay, they just assumed that the cell wasn't active then, it wasn't doing anything. So they just called it interphase. And they described it as a phase when the cell isn't really doing anything, which is nonsense because here the cells, the DNA is dispersed and I can get access to it and I can create proteins. Recognize during cell division, I can't make a single protein, right? I can't go to the nucleus here and say, okay, unwind for me and let me make a protein, right? So during cell division, I can't make proteins. The cell's at risk then. Here is when most of the stuff is happening. Okay, so interphase was called interphase because it didn't look like anything was going on. So they just said, this is the phase between cell divisions. And so what we're going to do here is we're going to put a pi here that goes all the way to the middle. And in the center here, I should have made this circle a little bigger, I'm sorry. In the center here, we're going to call all of this interphase. Okay, so all of this is interphase, and this is mitosis. Okay, so all of this is interphase, and this is mitosis. Interphase is usually longer than mitosis, and it accomplishes 3 purposes. Sometimes it accomplishes 4. The first thing I'm going to do is I'm going to recover from the previous cell division. Each daughter cell resulting from a cell division only gets half of its parent cytoplasm and organelles. The daughter cell has to spend some time replenishing its cytoplasm. Also, during that time, I need to start doing the things that I'm supposed to do. Okay? The cell isn't supposed to be doing anything during the time it's dividing, but when it's not dividing, it's supposed to be performing a function. So, for example, my skin cells, if this is a skin cell, okay, and I've got my nucleus there, the function of that skin cell is to create lots of protein in the cytoplasm that's tough and protective. What do we call all of these protein fibers in skin cells that are protective? I'm packing the cell with protective fibers. What's the name of that protein? It's also the same thing as the protein in my hair, and the protein in my fingernails.
Keratin?
It's keratin, right? Okay, so I'm spending a ton of time just creating lots and lots of keratin. Okay, so I'm recovering from the previous cell division, and then I'm also going to perform the function that I'm supposed to perform. Okay, and this kind of a skin cell won't divide again. It'll just continue to perform its function and perform its function. Okay, so we're going to call that G1. That's gap 1 phase, right? And in gap 1, we recover and function. Okay? If I were to put 2 words here, gap 1 is for recovery. Let's put recovery and functioning. Now, if I continue to function and I don't ever divide again, I stop dividing, I enter a phase that's called G0. Okay? I'm not going to divide anymore, so there's no need for me to continue with the rest of this. So I go into G0. No more division. Okay? No more division. A third function, if I am going to go ahead and divide again, I need to synthesize my DNA. So I call that the S phase. That's DNA synthesis. Okay. That's where replication occurs. And then the fourth function is to synthesize organelles and other molecules required for the next cell division. So that's my G2 or gap 2 phase. We're going to say synthesis for cell division, which is not DNA synthesis. I'm synthesizing everything else for my cell division. Okay. So I've described these things here. G1 phase, the cell has just finished dividing. The amount of cytoplasm and organelles is about half of what it was. During this phase, the cell recovers, completing most of its growth and synthesis of organelles and proteins. It also begins to carry out its function. This period can last for variable amounts of time depending on the cell type. Those that never divide never leave this phase. We send them to the G0 phase. S phase, when DNA is replicated to be ready for the next cell division. Usually once the S phase begins, the cell is committed to going through another cell division. G2 phase, usually the shortest phase. During this phase, many of the organelles and molecules required for the physical act of cell division are synthesized. When these phases are complete, the M phase begins. Okay? So, the thing about interphase is, while it sounds like a lot's going on, it appears that very little activity is taking place under the microscope. And before biologists understood that, they viewed the interphase as sort of a low activity phase where the cell was just sort of hanging out between phases when real activity occurred. Okay? So, I've got some pictures here. Okay, I've got real pictures, cartoons, and then another cartoon of the interphase state. So we need to go into some detail about what these things look like so that you can recognize it. So identify the set of pictures labeled interphase. This picture shows a diagram of a cell and an actual microscopic picture of the cell, and notice the following features. Okay, a discrete smooth border between the nucleus and the cytoplasm. What do I call that border between the nucleus and the cytoplasm? What? Thought I heard something. I call it the nuclear what?
Envelope?
Call it the nuclear envelope. Okay? Let's go look at that. Okay? I've got a nuclear envelope here. Okay? It's continuous and it's discrete. I can see it. It stains. Okay? Now I've got some pores in it, but I can't see the pores because that's too small for the light microscope. Okay? And then in this cartoon, I've got that nuclear envelope there as well. Okay? So that's the nuclear envelope. The blank in the nucleus is represented by linear or lace-like strands of darker purple material. We're going to call this the chromatin. And it's said to be dispersed throughout the nucleus. Okay, so the chromatin is this material here. Okay, and in some microscopic pictures, it looks like granules. Okay. In some microscopic pictures, it looks really granular. In other microscopic pictures, it looks more lace-like. And here, it's just depicted as being kind of lace-like or cord-like. Okay. It's a pair of blank lying near the nucleus in the cytoplasm. Okay, we can't normally see these under the light microscope, but if I've got my nucleus here, and I've got my cytoplasm out here in my cell membrane, I've got a pair of structures that looks like they're at right angles to each other. Okay, I've got a pair of centrosomes I've got a couple of these when I'm going to divide. Okay, these are star-shaped structures composed of come on, go back down here. Does anybody remember what the centrosomes or the centrioles are made of?
Calcium carbonate?
Not in, yeah, they're made of microtubules in humans. So the centrioles are these structures here. I've got two of the centrioles. This whole thing is a centrosome. Okay, and the reason they call it a centrosome is that S-O-M-E, some, refers to a body of some type, and this is the body that's central in the cell. It's central, it's sitting next to the nucleus, it's centrous, so they call it a centrosome. Okay, and these structures made up of microtubules are called centrioles. I've got an electron microscopic picture of this right here. So this is one of the centrioles, and now I'm looking down the barrel of one of the other centrioles. They're at right angles. And you'll notice that there's a lot of tubes surrounding it. A lot of these tubes surrounding it, these are microtubules, and the centrosomes, or the centrioles in the centrosomes, are the organizing center for microtubules. This is where the microtubules in the cell arise from. And the microtubules are structures that help keep the cell shape, right? They're like the rebar of the cell. Okay, so there's a pair of centrosomes lying near the nucleus in the cytoplasm. We can't see these in the microscopic picture of the cell. The stain doesn't provide enough contrast, and they're very small. If I look at the diagram of the centrioles, these are star-shaped structures composed of microtubules. Okay, these are filaments of the cytoskeleton composed of spherical proteins called tubulin. We'll talk more about that when we talk about the cell. The centrioles are centers of organization for microtubules. A pair of these structures lying at right angles to each other sits near one end of the nucleus during interphase, and microtubules appear to radiate outward from them. Again, the pair of centrioles is called a centrosome. Okay, a pair of centrioles is called a centrosome. I don't know how that cuts. Oh, somehow my picture got moved. Okay, at some point during interphase, in addition to the cell simply performing its intended function, the complete set of DNA instructions is duplicated in the nucleus, and then a second set of centrioles appears in the cytoplasm. So the centrosome, or the pair of centrioles, duplicates. So now I have two of them. However, under the microscope, we wouldn't be able to identify either event. The nucleus and the cytoplasm appear much the same throughout interphase. Okay, so even if this cell had already duplicated its DNA, getting ready for its next cell division, I wouldn't be able to tell that. It might be a little bit larger, but I wouldn't be able to tell that the DNA had been duplicated. And I certainly can't see the centrioles in the cell. Okay. So I keep moving my pictures. Okay, so in summary then, the goals of interphase are recovery from the previous cell division, synthesis of DNA for the next cell division, preparation of machinery for the next cell division. The nuclear features, and we'll follow these features through mitosis so that we can be able to identify all of these things under a microscope. I have an intact nuclear membrane, I have dispersed chromatin, and I have a nucleolus. Okay, I don't see a nucleolus in this cell. Maybe right here, there's a nucleolus. Here's a nucleolus. Does anybody remember what the nucleolus does? You'll need to know at some point here. What's the nucleolus do? What's that? Organization? No, not really organization. The nucleolus makes ribosomes. Okay, we have to have a ribosome factory. The ribosome is the protein factory, right? We have to have a factory that makes ribosomes, and ribosomes are made in the nucleolus. So it would make sense that if I'm going to divide, if I'm getting ready to divide, since I need to free up all of this chromatin into the individual chromosomes, my nuclear membrane has to break down, right? I don't need my nuclear membrane during cell division. It's holding on to the chromosomes, so I have to let go of them. So the cell membrane will disappear. I don't need to make proteins during cell division, so my nucleolus will disappear. And I need to condense my chromatin even further in order for these chromosomes to divide, so the chromosomes will appear. Okay, so if you learn to think about what happens in the nucleus because of the functioning of things, you'll learn to recognize the features of what is going to happen in mitosis. So you'll be able to describe those things. So in interphase, I need my chromosomes to be held in place, so I need an intact nuclear membrane. In interphase, I need to make proteins, so I need to make ribosomes, so I need a nucleolus. And in interphase, I need access to my DNA so I can read the code, so it's dispersed. Okay? And if you remember those things, you can remember everything else you need to know about the features of mitosis. Okay? So nuclear features: intact nuclear membrane, dispersed chromatin, and an intact nucleolus. And cytoplasmic features, I'm going to have duplication of the centrosome just prior to mitosis. Okay. So that's enough for one day. And what I'm going to ask you to do as a question of the day is be able to recreate this drawing. Okay, so when you come to class next time, I want you to be able to draw that from memory. Yeah. What's that? Yeah. So do the G0 as well. No more division. Just turn in the. You've got the. I know, but you've got the question of the day next time. I want you to have access. Just turn in the chromosome threshold. Wait.
No, the one I pulled out.
Oh, you finished that one. Yeah.
No.
So
We finished the one that we did last time today. 2 times ago, I spoke.
Oh.
We finished it, it's done. I pulled out the wrong one.
I see. I'm sorry. Don't confuse me.
Geez.
Did I? Oh, that was the. Did I not ask for that one?
No, it's okay. Walk through those lines. All right, we're telling them we're going to.
Did that thing?
No matter what the. Don't wait, that was really good. And the potatoes? What's the mashed ones?
Gnarly.
They're really good, actually.
This is all gnarly, right? What? I appreciate the question. I'm doing fine.
And they can categorize somewhere. Okay, I'm sacrificing the goodwill of everybody. I thought that was a dog. I was like, see ya. Yeah, I was like, oh, that's a different sound. Look, it's like ding ding ding.